Okay, let's go and get started. Today we are going to do surface area and volume. And another word that I don't want to say because I want to see if anybody in here can actually pronounce it properly without messing up. If you do mess it up, don't worry about it. We'll just point and laugh at you. Just kidding. Anybody try to pronounce this word? Here we go. Dan, how do you say that?
Microscopy?
Not microscopy, right? Microscopy. It's pronounced microscopy, and this is a branch of basically biology where you're actually using you're using microscopes to observe microscopic images. It could be research, it could be pathology, it could be surgery, medicine. You know, there's a whole division of surgery called microscopic surgery. I'm sure you guys have seen a lot of shows where doctors are working together and it looks like a big huddle. They're trying to put together a bone or trying to redirect blood through a heart. Well, there's also a branch of surgery where you simply have a doctor looking through an eyepiece. Like, imagine there's an eyepiece right in front of my face and I'm working down here on someone's body. And I'm looking through the microscope and the microscope is focused right where my hands are and where I'm doing all the work. Sometimes you might have a vascular surgeon. They might have to put together blood vessels, or a neurologist. They have to reconnect nerves. And some of these things are very, very small where you can't see them very clearly with the naked eye. So you want a more refined image. So we use microscopes. If we have time, I'll show you a couple of images of microscopic surgery. But we're going to do microscopy and surface area to volume ratios. But microscopy is one of those words where you really want to be sure that you're pronouncing it properly. There's a few other words you might have throughout the year that are a little tough. For instance, homogeneous, not homogeneous. There'll be a few others that show up. Sophia, where you at? Okay, so let's get to it. So whenever you use a microscope, the prefix micro means small, and scope means like to look or to see. Like on a submarine, they have a periscope. You want to see from far away. So this is all designed to see to make observations generally on the microscopic world. We're going to talk about 3 different microscopes today. And different microscopes can observe subjects in different ways, depending on which type of microscope you're using. Now, to understand the microscopic world, you need to understand how it's measured. And so for most of you, when you were in chemistry, you likely got a lesson in Unit 1 when you started that class on data analysis and measurements, and you mainly learned about the metric system. Well, I'm not going to rehash that entire unit. I'm just going to focus on a very select part of it. But the metric system is used by the vast majority of the world. In common measurements, we really don't use it here in the United States. If you have a contractor working on your house, or if you have a plumber and they're trying to measure pipes, or you have a contractor with wood and beams and all that, they typically measure things in inches and feet. Well, if you work at NASA, you work at SpaceX, those are American institutions. You work in hospitals, as TGH or Moffitt. Things are typically measured in the metric system: liters, milliliters, grams, meters. So you don't need to write this down. Don't write it down. I actually do write this part down, but the next thing you don't have to. What I really want to focus on are meters. And meters is a measurement of length and distance. Now, this is what you do not need to write down. Let's say that today after school, you're going to go out to Bayshore Boulevard. You're going to run a 5K. What does 5K stand for? 5 kilometers. So you see the word meters, which is what I'm focusing on, but kilo. You may have any idea what kilo means? Go ahead, Ryan. 1,000 times whatever the base unit is. So the base unit is meters. A kilometer is 1,000 meters. Well, when you have 5 in front of it, that basically means 5,000 meters. Or you could just say 5 kilometers. The other day, I saw on the news that the Coast Guard off the coast of Miami seized 500 kilograms of cocaine from a boat on its way to China or something like that. Well, 500 kilograms, how many grams are in a kilogram? Just by the prefix kilo, it means 1,000. So it's interchangeable between grams and liters and meters. You could do watts, you could do joules, you could do a lot of different things. So this would just be 500 kilograms. You add an extra 1,000 to that, that's going to be 500,000 grams. Because 1 kilogram is equal to 1,000 grams. But what I want to focus on is just the meters part. And we're not going to go bigger than a meter. I mean, we're talking about microscopy. We're going way, 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 way smaller. So there's about 1, 2, 3, 4. There's 5 increments of this I want you to know. First one is decimeter. Deci, the prefix D-E-C-I, deci, means 1/10 or 0.1 meters. It takes 10 decimeters to equal 1 meter. You obviously can see a decimeter with the naked eye. 1 meter is a little more than 3 feet. We like to say it's about the length of a baseball bat. So, you know, I'm 6'5", you would say I'm like 2.4, 2.5 meters. If you're 5'6" or so, we would say you're 1.6 meters, roughly, because a meter is about 3 feet. The next one is going to be centimeter. We're getting even smaller now. This is going to equal 1/100th or 0.01 meters. 100 centimeters is equal to 1 meter. A centimeter is about 2.54 inches. Or flip that. 1 inch is 2.54 centimeters. An inch is bigger than a centimeter. 1 inch is just about, and you hear that a lot in the metric system, you'll say, well, it's just about, when you try to convert a kilometer to a mile or a meter to a foot. A centimeter is, or an inch is about 2.54 centimeters. So an inch is bigger, and an inch is like that. So about 2.5 centimeters right there. Next is going to be millimeter. This is 1/1000th or 0.001 meters. 1,000 millimeters is equal to 1 meter. For those of you that know any Latin or Spanish, you know that cent or cien means 100, mil means 1,000. There's 100 years in a century. There's 1,000 years in a millennium. Perhaps I can help you with your translations. Those will eventually be on some of your Latin quizzes. You've already had centi. You'll see deci. You'll see milli. Now, I'm going to write a little line of demarcation here because now we're going to get microscopic. The next one is going to be a micrometer. And the symbol for a micrometer is not M, because M was taken from milli. The symbol that we use is the lowercase delta. I'm going to draw it really big over here. It looks like this. If you're familiar at all with Greek life, maybe you have a sibling, like a sister that's in a sorority in college. She might be part of the tridelts. Delta, delta, delta. That's the capital letter. It's like the Greek letter D. If, you know, with the hurricanes that we're all having this year, there's only one letter left in the Arabic alphabet, W. I think it's going to be Wilfred. After that, we have to go to the Greek. We got to go alpha, beta, and then eventually, you know, delta would be the letter D. So we're probably going to get to that. It's not even October, and we're going to have to start over again. Fun fact, that hasn't happened since 2005, where we've had to go into the Greek alphabet. Anyway, so from micrometer, this is going to be 1/1,000,000th, or 0.000001 meters. 1,000,000 micrometers is equal to 1 meter. And I'm going to write something here on the side. Generally, when we use micrometers, we're at the cellular plane. So there's different levels, and I'm going to perhaps show you this when I'm done here with these measurements. If you have to measure something with the micrometer, you're generally going to be about the size of cells, or something, or an organism like a bacteria or a microscopic worm that is really small. Micrometers. Okay, and the last one I would like you to know for our purposes here is nanometer. This is 1/1,000,000th, or 0.000000001. That's 8 zeros and then a 1 at the end. So we say 1,000,000,000 nanometers is equal to 1 meter. We generally use this for measuring molecules. If I want to know the length of a lipid or the length of a DNA strand or a protein, we would measure it in nanometers. So we have cellular, we have molecular. Anybody want to take a guess? What is one level smaller than molecular? Atomic. And then you go subatomic. And then you're watching Ant-Man. Quantum. Quantum, quantum, quantum. That's all very small stuff. So before I get into the types of microscopes, I think it'd be kind of cool to see just how small this goes. So there, when I teach chemistry, there's typically a website that I go to, and it's called The Scale of the Universe 2. I'm going to bring that up. And it really gives you an idea of just how big and small things in this universe are that you may have never known were actually there. So let me set this up real quick. We're just going to go through it for just a couple minutes. And it's blocked. Terrific. Maybe I can do Scale of the Universe 1. Well, that stinks. I guess I'll have to try to do it tomorrow. Let's try this. I think there's a YouTube video here. I can override that. Here we go. All right. So let's just get through this. We're going to skip the part that gets bigger. We'll go smaller. All right, so if we actually had this program, okay, we're going from big to small, so I guess we'll just write it out here. These are all the sizes of stars, planets and moons, states, countries. Okay, I'm going to slow things down. Okay, here we go. So this level right here, we would probably use decimeters and centimeters. Now we're getting pretty small. Coffee bean, pencil lead. Notice we're at millimeters right there. That's 10 to the negative third. Now we're getting really small. Pixels, grain of sand, dust mite. I'm going to show you an image of a dust mite using a scanning electron microscope. It's pretty gruesome. Now we're getting down to when we would actually use micrometers. We're getting down to the size of cells: skin cell, white blood cell, egg cell, cell nucleus, or chloroplast. Now we're down to micrometers. This is where you measure viruses. You measure perhaps organelles. And now we're down to nanometers. This is where you actually get down to the size of atoms, or not atoms, but molecules and things like proteins and DNA. That's as low as we could go for this class, but if you wanted to go lower, you just start shrinking, get even smaller. Size of protons and neutrons, quarks, which make up subatomic particles, and it just keeps going smaller and smaller and smaller and smaller and smaller, 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 smaller. And you're shrinking forever and ever and ever, and that's about as low as we know it goes so far. Okay, back to this. So maybe tomorrow I can get the Adobe Flash Maker working, Flash Player working, we can watch something like that. Okay, so let's talk about the microscopes that we have in mind for today. The first one is going to be a light microscope, or an LM. If you go to college and you guys want to go into biology or medical science of any sort, you're going to be using a light microscope during most of your labs. When I took microbiology, which is like studying bacteria and things, you have a lab once a week. You go to lecture twice a week for about an hour and 15 minutes. You go to lab once a week for about 2 hours and 15 minutes or 2 hours and 45 minutes, but it's just once a week. And the first thing you do, you drop off your stuff at your lab table and you go and get your microscope. And they are very, very delicate instruments. So the way a light microscope works is just what its name suggests. Light is shined through an object. And it is focused with a lens into our eyes. Now, thanks to Miss Rainbow downstairs, she let me borrow one of her light microscopes, and this is what we have at Plant. These are pretty small. They're not the best ones, but they do get the job done. You have to plug it in because it requires power for that light source. And right here, if you guys can see, there's light coming up from the bottom going up. You look in here, and you place whatever specimen that you want to observe, whether it be a cell, a hair, an ant, whatever. You place it on a microscope slide on this platform, and using these different power objectives, which I'll explain momentarily, you're actually able to view them at different levels or strengths of magnification. But you can't see anything if you don't have this light shining through it. And then the lenses that are in this eyepiece help focus the image so you can see it pretty well. These microscopes we have at Plant have 3 objectives. The ones that you get in college generally have 4. And I'll tell you how strong they can get right now. So in that eyepiece, 10 times magnification is already built in. For the ones we have here at Plant, and basically any high school in America for that matter, you have 3 objectives. Objectives are different strengths of magnification. We have 4 times, 10 times, and 40 times. But you have to remember that you already had the 10 times built in. So 4 times 10 is 40. So when you set it up to look at the 4 times magnification, you're actually going to be doing 40 times. 10 times 10 is 100 times, and 40 times 10 is 400 times. So you're definitely looking at things your eyes are not physiologically capable of seeing because it's too small. The only cell of the human body that you can actually see without a microscope is the human egg cell. It's just at the threshold of visible versus, I don't want to say invisible, but non-visible. It's the biggest cell of the human body. Now, if you were to go to college, there's actually a fourth objective. That's the 100 times objective. And you can actually see things at 1,000 times magnification, which is pretty cool. Now, with this, the image is colored. Sometimes you have to use a dye, D-Y-E. When I taught freshman bio, we did a lab with microscopes where the kids would take a toothpick and they would put the toothpick in their mouth and scrape the inside of their cheeks. They would then take that toothpick and they would twist the toothpick into a little puddle, a little drop of methylene blue. It's a type of dye. And that would stain their cheek cells blue and purple. So they would actually be visible when we put them under the microscope. Without that dye, you wouldn't be able to see them because those cells are pretty transparent. They're pretty clear. So a lot of times you have to have an image that is stained. So you can actually see things like see membranes and nuclei and all the other components. The specimen that you're looking at does not necessarily have to be dead. Like, is that an option? Yeah, most times with the other microscopes we're going to talk about, they have to be dead. For these, they don't. But here's the problem. The image is not very resolute. What I mean by that is it's low res. It's not, let's just say it's not HD 4K Blu-ray. So the image resolution is limited. And to show you guys what these look like, obviously there's a microscope right there. This is the eyepiece where you place your eyes. Some of them have two eyepieces. Here are the objectives. The light shines up from the base through the stage, and you have the knobs that can help you focus, help you with in the clarity. And then here's a couple images you can actually see. And I was actually very impressed with this image on the left. These are a bunch of plant cells, and if I try to zoom in even further, you can see their cell walls. You definitely know they're plant cells because they're very rigid. Cell walls are like body armor. They limit their motion and their size. And those little green discs inside are called thylakoid discs. That's where photosynthesis actually happens, within those discs. You'll learn about that in Unit 3. And then you look over here, these are the cheek cells that are stained blue and purple. The little grape-like structure is the actual nucleus. So it's pretty cool that you could scrape your own cheek cells, look at it under a microscope here at Plant, and you can actually see the nucleus. And you also know that your DNA is in your nucleus. So inside that is the essence of what you are. And then you can only see the plasma membrane. But what you cannot see are organelles. You can't see mitochondria, no Golgis, no ERs, nothing like that. With this type of microscope, if you want to see all those structures, you have to go even smaller. So now we're going to go super small. So the two microscopes you want to use if you want to look at something really tiny is a scanning electron microscope, or SEM. This microscope involves a beam of electrons. Electrons are the negatively charged subatomic particle in an atom. Okay. So Dan, I want to throw a hypothetical situation to you. You have a friend of yours that is invisible. They're not a ghost. You can't not walk through them. They're just invisible. So if they were in the hallway and you bumped into them, you would feel them. You just wouldn't see them. Got me? Mm-hmm. Now imagine that you want to see what this person looks like. So what you do is you have them lay down and you get a bucket of paint and you pour the paint on their body. Now, what are you technically seeing? Are you seeing their body or are you seeing the shape that the paint forms?
Their body.
But they're invisible.
You have to have a body.
But it's invisible. You can't see it. But I pour paint all over their body.
But
so
we know it's there because we poured paint on it.
You know that their body is there.
Yeah.
But because of the paint, you're actually able to see how they're shaped and their profile, their silhouette. So the way that this works, guys, is in this analogy, replace paint with electrons. The electrons cascade over the body or the specimen or whatever it is you're looking at. And because of those electrons, a shape is detected due to the sensors. So the sensors don't necessarily see the actual specimen. They see the shape that the electrons form by cascading over the specimen. Just like if someone was laying here invisible and we poured paint all over their body. So we could see every groove and bump and everything in their body. We could see where their nostrils are. We could see where their joints are and their fingers. We could see it all. So a few things with this. The specimen must be dead. If it was a living creature, it must be dead. Because you're going to shoot electrons at it. If it's not dead, it's going to be dead. The image is very high res, very good resolution. The image is black and white, B&W. So I'm going to show you what an actual scanning electron microscope looks like. And I'm going to show you some images that it takes. Okay. So this is a scanning electron microscope. Typically you have this, it looks like a tall cylinder. It's roughly about 3 feet high. And generally right around here, you would have a computer that the image would be projected onto. And what happens is up here at the very top is the source of the electrons. The electrons are focused down, and then they're going to be shot at whatever specimen is that you're looking at. And there's a sensor inside that's going to detect where all the electrons scatter to. And that's going to help form an image. And the images are actually really cool. So this is the face of a parasitic worm. And if you look really closely, that line represents 50 micrometers. So we're getting very, very small. And so some of those worms that you can get from, dogs get them, people get them, parasitic worms, but these are nearly microscopic worms. You wonder, how can they do so much damage? Well, this is how. There are some parasites that are passed from species to species, and some of them can eat your eyeballs, they can eat your brain, they can go throughout your body. And you're like, well, how can a little worm do all that damage? Look at that. It's pretty sharp little teeth. Over here is the face of a butterfly. You see the eyes. Look at that high-res image. There's the tongue right there they use to drink the nectar out of flowers. And over here are some red blood cells. Now, this has been photoshopped because, as I said, scanning electron microscopes cannot see colors. So somebody just took that image and they photoshopped it to make the red blood cells red. Yeah.
Do they generate their own electrons, or is it like?
Well, it has a power source, so you can get electrons from electricity. You just have a positively charged magnet that attracts the oppositely charged electrons, and that just focuses them and shoots them. Now, is anybody here arachnophobic? So if I show a spider, are you going to freak out? Okay. So there, you can look at a lot of cool images if you just were to type in. I know you guys do this in your spare time all the time. You go to Google, type in scanning electron microscope spider. Look at that face. How you doing? Grimace from McDonald's. Interesting. Look at that. Pretty cool. Does anybody know what a tardigrade is? A water bear? So let me see if I can pull that up. Their nickname is a water bear. There they are. These things are bulletproof. There's been 5 mass extinctions in Earth's history. They've survived all 5. They can live in space. One of them almost ate Ant-Man. Oh, here's one more. This will make you face mite. This lives on your faces. Where's this? There it is. Hey, babe. There are tiny little skin mites that come out at night to have sex on your face. That's why you need to wash your face. Ha! All right, moving on. Isn't biology dirty? Okay, next, last one is we have transmission electron microscope, or TEM. Now, for those of you who don't have a super short-term memory, when you have a scanning electron microscope, when the electrons hit the body of your specimen that you're trying to observe, what do the electrons do? Ryan? They cascade over it. Transmission electron microscopes shoot electrons right through it. So in this case, high energy electrons are shot right through whatever it is you're trying to look at, and it projects an image. Now we use this one to look at generally at the inside of cells that we can't see with the light microscope, because the light microscope is not, it has a limited resolution and a limited magnification. Generally, light microscopes can go a little more than 1,000 times magnification. These things can go like 100,000 times magnification. So we can see things like organelles, we can see viruses, we can see molecules like proteins. In fact, the coronavirus, it's called COVID-19, not because it's the 19th coronavirus. Some people think that. It's because it was discovered in 2019, in about November of 2019. And it's a whole class of viruses called coronaviruses. This is just one that we're dealing with now. And the way that we found out, or that it got its name, is because of scanning electron microscopes. What they learned is that the virus has a capsid, which is a protein casing. And on the protein casing are these little projections. And when people saw images like this, it reminded them of the projections on a king or queen's crown. And for those of you that know any Spanish, what does corona mean? It means crown. So that's how it got its name. Crown virus, which if you actually translate that, it means crown poison, because virus means poison. The images are black and white, very high res. Specimen must be dead. And you're generally looking at the inside of a specimen. That's why you have a transmission microscope rather than a scanning, because transmission goes right through it. And again, we use this to look at organelles, we look molecules, inside cells. Yeah.
So shooting the electron through the thing, does it cause any chemical reaction or anything, right?
All right, so I'm going to show you some images of this. This is a scanning electron, or excuse me, a transmission electron microscope. You can see it looks to be about 3.5, 4 feet tall. It's on that desk, and you place the specimen inside that cylinder, and the electrons are shot right through it, and an image is projected on the computer. And here are some of the actual images. So this is pretty cool. This is a cell. There's the cell membrane. This is the nucleus, and you can actually see the DNA inside the nucleus. You cannot do that with a light microscope. Over here, you have a cell with all the various little goodies inside on the right. And then down here, these are mitochondria. If I look closely, you can actually see the folds inside. It's called cristae. That is a very, very resolute picture for something so small. That's probably more than 100,000 times magnification. Very tiny. But they are down there. If you actually look, you can see the measurement is about 50 nanometers across. That little black line down there is 50 nanometers. So that's maybe a couple hundred nanometers for the entire mitochondria. So these are very, very high resolution images for stuff that is so small. Pretty impressive. Okay, so that's everything with microscopes. Now let's get to the second part of our lecture. And we have about 8 minutes, so I'm going to try to get through all this. Surface area to volume ratio. Okay, I want to give you guys a size comparison. This room is room 130 or 232. And then we have the gym. Here's a door, here's a door. For my room, here's a door, and there's a door. There's a back door back there in the Miss Smith's room. Which room would it take longer to walk through? The gym or room 232? The gym, and it's not even close. You have to go all the way through the other side. It could take a long time. As in my room, it's smaller. So my room has a greater surface area to volume ratio than the gym does. And this is very important to know with cells. Cells are very, very small structures, but we need to know why. So this question is going to lead me to my point. Why are cells so small? The answer is efficiency. We have innumerable substances diffusing into and out of our cells all the time. You have glucose, you have other nutrients like food that your cell needs. You have vitamins and minerals, you have oxygen and carbon dioxide. They have to be able to be exchanged into and out of the cell quickly because they're needed. But if it takes a long time for oxygen and CO2 to diffuse back and forth through a cell, that could actually jeopardize the metabolism of the cell, which could affect the health of the cell. So the smaller the cell, the more efficient it is. The smaller the cell, the greater the surface area to volume ratio is going to be. So to understand what I'm getting at here, remember that the greater the number, the more efficient it is. And someone last period said, what do you mean by efficient? I mean by its ability to have important materials diffuse into and out of the cell. If you have a big cell like the gym, it's going to take a long time for things to go in and out because the gym is so big. But if you have a room like this one, it's not going to take long to go in and out, and so you can be more efficient. So let's say that we have a cell like this, and we're going to calculate the surface area, and we're going to calculate the volume. The volume, or excuse me, the surface area, we have 1, 2, 3, 4. So the surface area is 4. Well, inside the volume is 1. So we would say the surface area to volume ratio is 4 to 1. Guys, you always want your volume to be 1. You always want to say 4 to 1, 2.5 to 1, 10 to 1, whatever it is. talking all day. Voice is finally getting sore. So let's check out this cell. Look at this. We have 4 cells together. Make something bigger. Surface area. 1, 2. Oh, wait, wrong color. 1. There we go. 1, 2, 3, 4, 5, 6, 7, 8. Surface area is an 8. Volume. 1. Come on. 1, 2, 3, 4. The volume is 4. We have an 8 to 4 ratio. What does that reduce to? 2 to 1. So I ask you, what number is bigger? 4 to 1 or 10 to 1? Or sorry, 2 to 1. Really? 2 is bigger than 4? No, it isn't. 4 to 1. What cell do you think, just use your own eyes, no tricks. What cell looks like it would take a longer time to walk through? The first one or the second one? The second one would take longer to walk through, so things are going to take longer to diffuse through it. That's going to be less efficient. This is better than this. This is going to be more efficient. Let's do a couple more examples of this. What if you have something like this? One, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4. Thorson. Okay, I'll send them down. Sage Stark, you're signing out. The surface area is a 10. The volume is a 4. What is 10 divided by 4? 2.5. So the surface area to volume ratio is 2.5 to 1. It's not bad. It's not as good what we had to start with. And then the last one, guys, we'll try this one. 0.5, 1.0. 1.5, 2.0. 2.5, 3.0. 3.5, 4.0. So that's the surface, or excuse me, the volume is going to be a 4. Surface area, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Surface area is 18. So you do 18 divided by 4. 4.5 to 1. That's pretty good. That's better than what we started off with. That means that things can really diffuse through that cell pretty fast. So the greater the number, the greater the surface area, which actually is hard for your brain to wrap around this, the smaller the cell is. But that means it's more efficient. Things can diffuse into and out of it much faster, and that's the name of the game. You don't want a cell to be too big because then it's going to take a very long time for oxygen and carbon dioxide and glucose and other water to go through it. And if it takes too long, the cell could die because it's not getting what it needs in a timely manner. All right. There was a few other things I wanted to get to, but we'll have to put it off to next time. Tomorrow we're going to do the plasma membrane.